Thank you for coming, and the MICCAI Society is very excited to announce a series of scientific and educational talks from MICCAI Fellows. I'm very glad to introduce the first talk of this series featuring Professor Alex Frangi, a dear friend and a prestigious scientist at MICCAI Society. Let's start the first talk featuring Professor Alex Frangi of the MICCAI Fellow Talks. So Alex, the time is over to you now.
Super, Jai. Thanks a lot. And it's really a pleasure and a privilege to be able to talk today to all of you. And I'd like to talk about the work that we are doing in Leeds on computational precision medicine, and in particular the work we are doing around in silico trials. So this is work that is been undertaken with funding by the Royal Academy of Engineering in a 10-year award that has the focus of promoting new emerging technologies. And the work that I will present is actually the work not only of colleagues, definitely is not only my work, but it's the work of also other colleagues that I'm working with in Leeds that I will introduce, and also of past collaborators in Barcelona that were part of my group when I was there. So what I'd like to do today is I would like to focus, given that I'm talking, let's say, to colleagues from my own community, from the medical image computing community, I felt that it was more interesting if, rather than going and presenting a number of algorithms of image analysis, with some levels of novelties in those, that what I would present is more like the vision behind the work that I'm trying to do and the connections between that work in medical image computing and other disciplines, because I think that, that could be a stimulating of new ideas, but most importantly, can explain a bit how some of us at least look for inspiration in other neighboring fields. Beyond, of course, medicine that we all work with. So what I call the story behind the story of the work that I do is essentially the development of models from both disease and therapies, building them from individual images, create models that represent populations, and then utilize those models to go back all the way to develop novel medical devices, whether it's imaging techniques or implants. Similar ideas can be applied as well to drug design, but that's not an area where I personally work, although there is quite a lot of other colleagues in that area. So this is a vision that we've been developing for a number of years with very close colleagues in Leeds, with Sig Taylor, Tony Lassila, and Nishant. So I'm an electrical engineer by training, Zig is a mechanical engineer, Tony is a mathematician, and Nishant is a biomedical engineer. And you see here how interdisciplinarity is actually quite critical. So if you look at what's going on in medicine, and this comes from an IBM report from just a few years now, where we were looking at the importance of the access to patient data, patient information, but also the access to relevant clinical knowledge. And you can imagine this in two different dimensions. I tend to think about this as a sort of a sat-nav system, where on the one side, what we have is the opportunity to do GPS measurements with a lot of accuracy, which is basically the ability to extract, for instance, imaging phenotypes and other phenotypes, and then the ability to develop manifolds or learned representations for regions, which is basically building charts. And in a sat-nav, both things combine. You can imagine how useless a sat-nav would be if you have very nice maps but no position, or if you have the position but no map. And I think in health happens the same. We need to understand both the position of a patient that we're trying to diagnose or treat, but also we need to understand how that maps to the wider population. So what computational medicine is all about is combining techniques from imaging, from mathematics, from computer science, and from engineering to actually improve our understanding on health and also the treatment of disease. And we look at that both at the individual level, but also at the population level. And I think one of the areas that excites me, particularly in the medical image computing domain, is the possibility to scale a lot of the things that we've been developing over the last 20, 30 years to really very large cohorts. And the sort of question that I seek to answer in this area is what constitutes the pattern of a normal anatomy or function, how you quantitatively describe disease across the population of subjects, how you objectively identify subjects at risk, so subjects which are kind of in neighborhoods within our map that are dangerous, how you develop optimal treatment strategies for a given individual. So what are all the things that are in that neighborhood of that patient that would make the patient respond better? And of course, what you can invert the question and say, what are going to be the patients that will benefit the most from a given device or therapy, which is a way to say, you know, what is the space of influence of that particular device and therapy within that chart. So a lot of these ideas, if you want to read more, we put them together in a number of editorial papers and viewpoint papers over the years that summarize a lot of these ideas. But one of the key things that I think could be helpful is to understand what is called the DAKY pyramid, or data, information, knowledge, and wisdom pyramid, that essentially is an abstraction already from the 60s of the last century, from information sciences, where the idea is that, you know, yeah, we have lots of data, imaging, and other sort of medical data, but that's actually not very useful unless you're able to structure that around in patterns, and therefore deliver information, whether you actually take that information into context and in a synthesized way. And that's what we call knowledge. And then when that knowledge gets validated over a number of, you know, cohorts, then that's what becomes wisdom or common understanding. And wisdom in medicine is equivalent to clinical guidelines, for instance, but also in the image computing domain, you know, wisdom becomes, you know, that experience that we have, that certain algorithms work better than others in a consistent and systematic way. Now, only when we actually have wisdom is that we can really start looking at future actions, because everything we have from data to knowledge is mostly a synthesis of the past, of the observations that we have. But what you really are interested when, or a clinician is interested, what is in front of the patient, is to decide what to do with Mrs. Smith, right? Now, there are two ways to do this, to go from data to wisdom. One, or at least two ways. One is what we call bottom-up phenomenological data-driven approaches, and that's where deep learning to a large extent lies. And then mechanistic approaches, which are more top-down, model-based, driven by priors and prior knowledge. And these two ways of doing artificial intelligence, the phenomenological and the mechanistic, I think are actually quite important and are starting to deliver a lot of interesting insights. And then there is combined versions that I call the middle out, where you start from, you know, you never quite have both you know, pure options. So the first phenomenological approaches are great when you have no understanding on the phenomenon of the mechanisms, where you have lots of data. The mechanistic ones, you may have limited data, but you understand the fundament, you know, the foundations of the physics, the physiology, the anatomy, and you can put that in the form of priors. Now, many of us may have the impression that with current you know, advent of artificial intelligence and AI and deep learning and other techniques, there is no need anymore to do theory, because you can always train things. And I strongly believe against this view. I think data-driven approaches are very useful, but I hope I convince you by the end of this talk that this is not enough, and that there is a number of other people who also think the same way and believe that there is a space for you know, more sort of principles or mechanisms-driven approaches. So let me give a first step to an example about why that is the case. So this plot comes from a colleague in ETH, from Dirk Helbing, who is a computational sociologist. And he comes from theoretical physics and has developed a number of models for understanding societies and complex systems. And he has a very nice diagram where he shows that as we evolve over the next few years, computational power will increase, as we know, through the Moore's law, and that doubles every 18 months roughly, and it follows an exponential growth. There is also evidence that data is increasing at exponential rates with a rate that actually is also in itself increasing. So currently is about doubling every 12 months, but people talk that in a not too distant future, data will very likely double every 12 hours. So but it's exponential. Okay, so what that means is that rate is faster than the processing power. So very quickly, if not already, we're in a situation where there is more data we can possibly compute. And then the most interesting bit is actually that the systems we're trying to understand, and by systems you could imagine the neural networks that we are trying to train that have an increasingly larger number of parameters, but also the disease processes that we're trying to understand, especially when you start looking into comorbidities and interactions between disease. Those are complex systems that have been modeled having a factorial growth of demands of complexity with time. So you can see how very, very quickly we will be in a situation where systems we want to identify are way more complex than data we will be able to collect to inform those systems, and let alone, you know, much bigger than the computational power that we will have. So in order to break this course of you know, dimensionality scale, you could call, I think the only way to do that is by introducing priors. And this is why a lot of people, even in the machine learning domain and in the deep learning domain, are starting to look into things like physics-informed networks that actually try to incorporate some of the priors in ways of regularizers, loss functions, you know, part of the architecture sometimes. Okay? And the minute that you talk about introducing priors, you need to go back to the theory and you need to understand what you're doing. It cannot be purely 100% data-driven. So what I do, and we do in our team, is this interplay between these both type of models, and we talk about image-based personalized modeling, but also model-based predictive imaging. So using information about the patient to generate markers that are image-like, so spatial distributions of parameters that you couldn't potentially obtain purely on the basis of an image acquisition. And both of them will require is an interplay of information processing and knowledge modeling. And that's why processing and modeling are core, in my view, to the future of computational imaging. So just to give you an example, I'm sure many of you are familiar with model-informed or physics-informed machine learning. These are examples where your neural network also has a loss function, where the loss function represents the physics, for instance, of the process that you're trying to emulate. Imagine that you're trying to put as an input geometry and to output flow in vessels. You can do that in principle with just, you know, pairs of geometries and flow patterns, but then nobody guarantees you that when you are given a new geometry, the flow that you will generate will satisfy, for instance, the Navier-Stokes equations. So by imposing that through the loss terms, you can actually ensure that through your learning, you only have solutions that are compliant with the physics, you know, that needs to be guaranteed, or the conservation laws that need to be guaranteed. And you know, a good friend of mine, Andreas Meyer, that many of you will know from Erlangen, wrote you know, a couple of years ago, I think now, a paper in Nature Machine Intelligence, which essentially he gives a bit of the foundations for this problem. He talks about known operators, but that's another way of saying model-based information. And he demonstrates that if you incorporate known operators in deep learning algorithms, effectively you reduce the error bounds, because effectively what you're doing is injecting priors. And that, of course, helps your predictions and your inference. So over the last few years, we've been progressing more around the application of a lot of these ideas to medical devices and medical implants. And now we you know, also with involvement from Seppo Virtanen, who is another colleague in Leeds, who is a statistician in our group. And what we got inspired is by the fact that if you look at something which is called the implant files, which is an investigative report from investigative journalists. They looked over the period of 2008 to 2018 to all the medical devices failures from medical devices that had FDA approval. And what they observed is that just in that 10 years of time, there was at least 83,000 deaths reported for failures of medical devices, and over 1.7 million devices, failures and serious injuries as a consequence in people just in the UK alone, in the US alone. So there is a serious issue here, because if our regulatory process that should, you know, ensure safety is still allowing all of this to happen, is that perhaps we can do things better. And this is only going to get worse, because as technology becomes more and more complex, the rate at which we are able to innovate and come up with new methods of implants is faster. So the amount of things that we need to regulate is faster as well. Devices also are more convergence in the technology. In the past, a medical device was perhaps a hip prosthesis, which was a metal implant. But now those implants also may have a coating. They may have sensors that emit signals that then some AI does some analytics, outside the body. And those systems become very, very complicated to evaluate. That increases the regulatory burden as well. We've seen that there is serious adverse effects, and that reduces trust in the industry sector and shareholder value, with implications as well that regulations become even more bigger, more burdensome, and that increases, R&D cost, time to market, etc. So this needs to change, and basically that means that we need to go back to basics and see, what is the life cycle of a medical device product, and all the different phases, from the idea of a new device, the R&D you need to do, the feasibility, then the clinical trials and all the post-market surveillance that you need to do, and then go back to revise your ideas when you understand that there are problems and loop. The problem is that this loop at the moment takes easily 7-8 years when you talk about a medical device, and more like 11 to 13 years if you talk about a drug. So it's a very slow process, that in the process, there's many people who are being either not benefiting from new therapies which are going through this process, or people who are suffering from the side effects and the failures of therapies that, in a post-market analysis will be seen that they are problematic. So the value proposition, or the aim in our group, is actually reduce this R&D from years and to months, and reduce the risk by failing fast and failing earlier. Just to put things in context, the pharma and the medtech industry are growing at 5 to 6% annual growth rate over the last 20 years. However, the output from FDA approvals are flat over the last 20 years. So it means that there is more demand and there is more products, but actually we are killing a lot of those products at the regulatory phase or earlier on. So the system is very inefficient and with a huge attrition rate. How do we demonstrate safety and efficacy of devices? Well, the sources of regulatory evidence are either animal testing, bench testing, and human testing. And what we are advocating is the opportunity to use more in silico or computational techniques as an alternative to all of those. And that's what most of the rest of the talk will be about. If you look at other sectors, like for instance, the automotive sector is about 4 times larger than the medtech devices. And over the last 20 years, we've seen massive improvements in the products, but also in the process by which we develop those products. We don't do wind tunnels anymore. We tend to do aerodynamic simulations. We tend not to do crash tests, physical crash tests, but we can do virtual crash tests. We can do virtual prototyping, and we talk about AI self-driving cars. So why couldn't we be doing similar things in the medical domain? So this is an area where I'm working for the last 15 years, which is brain aneurysms, and I use it as an exemplar that has all these issues that I was describing before. So what I'm showing here is the evolution over 30 years of medical implant from brain aneurysms. And what you see is that ultimately it's still a bunch of wires, okay? Then we have a still 20% complication rate and nearly 3% mortality rate. And we could do the same sort of simulations that we do for the aerospace and the automotive industry. So why cannot we do the same in here? Well, that's basically what in silico trials is the idea of being able to reduce the need of conventional trials, to refine those trials that we do, because we can estimate the effects and we can then do proper sizing, and to replace those trials there where it's impossible to do it, or they are unethical to do it, or where there is, or where in silico is the only way to go into a more inclusive approach. And the vision is to go from this process where we go from bench, animal, and human testing to a situation where we still need at least some human pivotal trials, but where we have a larger amount of virtual patients and virtual populations and computer-driven models. So, okay, what does it mean in practice? And what does it mean for people like us that do medical imaging? So imagine that you could get your hands on a very large database of any particular organ system you're interested in, and you can build libraries of digital replicas, digital twins of anatomical models of interest, in this case, brain aneurysms. And this is just an example of a much larger population we have. Now imagine that what we can do as well is somehow virtually model the process, well, the device, the stents in this case, and the process of deploying this stent in each of these anatomical models. And imagine now that we can model the fluid dynamics within those anatomies. And imagine that we can develop now some metric of clinical outcome, that in our case is the reduction of flow after having put the stent. So after we put the stent, we will reduce the amount of blood that goes into the aneurysm. We will slow down the blood. What we will produce is a coagulation process that will close the aneurysm. So we want to reduce the amount of flow that goes. We want to slow it down. So from that perspective, this device in this particular patient is performing better than the same device on this different patient that has a different anatomical configuration. Now imagine you did that, and you did that over the whole set. But now actually you could say, well, but what were the physiological regimes of those patients? Were these people at rest or were they in exercise? So you can now do the same patient on this with the same device, but on multiple regimes of physiology, and again look at what is the flow reduction. And of course, you can then go and put different devices and look at what happens when you test multiple implants in this case. Okay, so the way I think about an in silico trial is like this massive ensemble simulation space, which has multiple hyperdimensions, one where you have the geometry, where you have mechanical properties, where you have rheological envelopes. And we need to build fundamentally representations and manifolds for representing those very complex, abstract objects. And imagine how you develop a generative model that can model vascular trees with, varying topology. But then also how you do that in a way that is not just the geometry, but is the geometry, the mechanical properties, the rheology, in a way that all of those cross correlations are consistent with, plausible anatomic human behavior. You also it's not just about imaging. You also need to look at, for instance, signals, and do the same with lifestyle and physiological envelopes, but also operational envelopes. Imagine that, our community has a lot of people working on the MICCAI domain and computer-assisted interventions. And for those of you who work in that area, the operational envelope could be the uncertainties you have in terms of the operator who is inserting this device, and the limitations you have in guiding that through imaging, for instance. So how you model and capture that, and how you also model the devices themselves or the interventions themselves. So here is where things also start becoming exciting and connected with these larger scale, data analytics or larger scale image analytics, because in order to create those virtual populations of anatomies or physiologies, you need to be able to analyze at scale data sets which are population studies or real world data in the first place. So let me show you now a case study from this vision now a bit into action, so that I illustrate a bit what we've been discussing. And I'm going to do that, how not, in the aneurysms space. It's not the only area we're working with, but this is one where we have quite a lot of progress being made over the years. So let me recap a bit about what aneurysms are. So aneurysms are a bulge in the vessels. They like to happen in bifurcations. They like to happen in the brain, and particularly around the circle of Willis. And when they happen, and if they rupture, they're lethal. A third of the people die straight away. A third of the people have high comorbidities long-term, and a third make it, to continue in a healthy way. There are different ways to treat them. The very old way of treating it was by putting a clip that requires brain craniotomy and putting a clip at the neck of the aneurysm. And then we have the minimal invasive approaches, which all of them are under image guidance and going through an incision in the groin, all the vessels up through catheters. And then you have coils. You also have coils. If you have wide necks, you need to put some sort of a stent to hold them, so act as scaffolding. And then people say, well, if we're putting a stent, why don't we put a stent that is very porous, so that actually reduces the amount of flow going in the aneurysm, and we don't need the coils in the first place. Now, do you remember the point I made about Dear Helving, that the systems are complex? Well, this diagram is a simplified version of what's going on in relationship to the rupture of aneurysms, and how aspects connected with the wall tension, the hemodynamics, the geometry, the wall structure, the thrombosis, the perianeurysmal contacts, and aspects connected with, the genetics, the physiology of the patient all interact. And that's why this is growing in a factorial way, because all of those interactions are everything with everything potentially, right? So what we did in the past is develop techniques that actually allowed to analyze patients' phenotypes in detail. This is a bit like the GPS position that I was talking before, and analyze things like, for this particular patient, how different approaches to treatment would affect the life expectancy from all of those patients. Like if you don't treat this patient and you just have a watchful waiting, the expectancy might be 8 years. If you do a clipping, then you increase the life expectancy, but you also increase the risk at the time of intervention because you're making an invasive procedure, and so on and so forth. So the idea here was to do risk balance decisions around particular individuals. And we also worked in the past with Philips to develop enhancements driven by models of their imaging platforms, so that you can actually do simulations and you could do planning, for instance, on a patient-specific manners. So what you have in the end is a complex information processing and knowledge modeling pipeline, where you have these pipelines that go from images to do segmentations, then do surface correction, generating volumetric meshes, setting boundary conditions, solving Navier-Stokes, and then you basically spit out these complex visualizations, which are virtual images. That's what I was talking about, virtual imaging. This is a virtual imaging of a streamlines, of oscillatory shear index, of wall shear stresses. These are quantities that are hidden. We cannot directly measure them with imaging, or at least not with X-ray. But if you then do that in a repetitive manner over a large set database, then you start understanding the patterns of how anatomy, shape indexes, hemodynamics, chemistry, all of those things, essentially how all of those things interrelate to each other, and what patterns are formed that tells you what patients might be at closer state to risk. Now, what is the difference between all of that past work and work that we all do, we continue doing in this area, with in silico trials? Well, in silico trials to me has 3 main challenges, and I'm borrowing here a slide from some colleagues in Duke that capture that, I think, in a beautiful way. So on the one side, we want to do virtual patients and virtual populations. So we want to go from images, but also other data, to these digital representations of the human. We also want to do virtual models of the interventions, the stenting, but also of examinations, the imaging process. So don't think about in silico trials only useful for implants. It is also useful if you want to develop novel imaging biomarkers. I think personally in the future, we need to move away from this view that we work with clinical colleagues where they give us a database with normals and people with a particular disease. We throw our pipelines for radiomics or deep learning. We find patterns and we try to learn some boundaries, and that's the way we discover biomarkers, to a more constructive rather than deductive approach to biomarkers, where we actually, biomarker engineering, where we actually solve the forward problem of the image acquisition. We model, to the extent that is possible, the physiology and the disease process, and we then essentially tell ahead of time how the image would look like for a patient that has a given severity of disease. I think we will get a stronger hold on the imaging as a technology doing that way. I think I'm sure we all have that experience, that feeling sometimes, that the way we do it at the moment sounds a bit as a fishing expedition in many occasions. So let's look now at some of the underpinning methods and for that application. So I talked about the devices. This is just a picture now, so you see how they look like, the various devices I showed you before. I also talked a bit that the reason why we put these devices is to slow down the flow, because if we slow down the flow, what happens when you have a wound that is bleeding, you put your hand to actually try to create a tampon with it and reduce the flow, but also stagnate the blood, and that creates a process of coagulation. And that hopefully reduces the model with time. Devices are very complex. I only show some aspects in the simulations that I showed you before, but they are potentially very complex. And let's also not forget that the devices can fail not only because the device itself fails, but also because of the delivery system to bring that device in place fails, or because of the image guidance procedures that we have fails. So there is a space to do evaluation at every stage. So let me show you, to give you a bit of a sense of scale of the things I'm talking about. So this paper that you see here is a paper in a very reputed journal that reports on what is called the PUFS trial, or pipeline for uncoilable or failed aneurysms. Pipeline is actually the name of a commercial device. It's called PED, or pipeline endovascular device, and it's a device from Medtronic and from Covidien before, an EV3 by the time the study was done, but it was bought by Medtronic. And this device, they claim that is one of the most studied devices for brain aneurysms. There is tons of these devices for treating them, but this one has several studies. And each of those studies, which I'm going to talk primarily about PREMIERE, ASPIRE, and PUFS, which are the three biggest ones, if you put the three of them together, the N, that's an add up to 500 patients. That's all the evidence they had before they went to put this in patients. This publication alone, which is one of the trials, took about 8 years from design to publication, 6 years of patient recruitment, only to collect 108 patients. And this cost probably between 20 and 40 million. And that's one trial, and there are 3 of them, okay? So what I'm going to show you now is what we call FDPAS, which is an in silico trial that was benchmarked and compared against the 3 trials from that device PED. And what I want to show is that we managed to get accuracy to a 5% of the same endpoints. We could run this trial, assuming you have all the modeling tools and everything in place, within less than 3 months. And it costs a fraction of that cost that the other study costed. So what we did is we created a pipeline very similar to the one I showed you before. But the only thing I didn't tell you before is that in addition to model the fluid dynamics, we also modeled the coagulation of the blood vessel. And this means that you're also adding, biochemistry in addition to the physics. Now, we didn't have access to the data from those 3 trials. So what we had is we worked with a data set from a previous European project that I led a number of years ago. And what we demonstrated is that we could create that population of aneurysms we had the same demographics in terms of typical parameters that clinically are considered, like neck width, maximum diameter, than the conventional studies. So PUFS, ASPIRE, and PREMIERE, and our in silico trial. And these are all the demographics in terms of age, sex splits, mean diameters, mean ages. If you look at this in detail, fundamentally, there is no statistical significant difference between all of those populations. The other thing they use as effectiveness endpoint was what is called the angiographic occlusion, which is the fact that after they put the aneurysm, the stent, they flush contrast agent to actually see how much contrast gets into the aneurysm itself. And if you don't see contrast getting into the aneurysm, you regard the aneurysm as angiographically occluded or closed. And that's a measure of success. You will see that sometimes we talk about 164, sometimes we talk about 88. The reality is there were about 88 aneurysms. Some of them, all of them were double, so we had both normotensive and hypertensive conditions. These we can simulate for each of the 2, for each anatomy, we can simulate like 2 patients and the 2 different blood pressure regimes. And then there is some of those that had to be removed because of failure of algorithms. So it's like equivalent to a drop-off rate in a conventional trial. So what you see here is that the predicted angiographic occlusion with the in silico tools was 75% overall, while it was also about 75% in each of the other trials. In general, we tend to be good at predicting short-term outcomes, so up to a year. The problem is that some of these patients, as you follow them up over time, there is a different success rate, and some of them tend to clot later, and longer-term predictions tend to be a bit more challenging. Now, the other thing you might see here in sort of somehow downlighted is that there is an 83% success rate for NT, which means normotensive patients, and a 67% for HD, meaning hypertensive. What it means is that if you don't consider the level of pressure, you will end up with an average value of outcome, which actually is not representative of very clear patient populations that exist in reality. And the fact that we are doing this in a computational manner allows us to explore in a lot more detail subpopulations, like, for instance, aneurysms that have hypertension, aneurysms that have side branches, aneurysms that have giant and large aneurysms, and other situations that in conventional trials you will have to have thousands of patients to have sufficient statistics to estimate those. And then the other thing which is quite interesting is not only that we can replicate what a conventional trial does, you can actually go beyond what a conventional trial allows you to do. And let me show you as an example of that this particular case. So case 3 and case 4, if you look carefully, the anatomy is the same. It's the same patient. The difference is that, well, the same patient, the same anatomy, okay? But we simulated a normotensive patient, so putting boundary conditions from normotensive conditions and a hypertensive one. And what you observe is that in the hypertensive case, in the normotensive case, the aneurysm clots, which is fine. So this color coding represents the clotting density, the clot density. And you also see that there is a side branch that is okay. It still has a good patency. In the hypertensive case, however, what you observe is that this aneurysm, as the clotting, the time goes by, the clotting is not as fast as the previous one, but also you start getting clot into the side branch, which is very, very dangerous because you will produce a ischemic stroke downstream. Okay, so that and that's something that there is some anecdotal evidence that was happening, but there is no systematic understanding about why that happens. And what this approach allows you is to study in a much more thorough way your space of solutions and understand the potential risk. So you can then develop devices which perhaps are more effective in avoiding that. So what next? What are the underpinning methods behind a lot of these tools? So, and again, I'm not going to go through telling you how to segment vessels and stuff that we all know and we've been, many of us doing either PhDs or careers on, but what I like to explain is some of the challenges that you can be forgiven about those when you're doing purely diagnostic applications, but when you want to do in silico trials, they become very, very challenging. So I'm going to focus on a few ideas only. Imagine you want to build these virtual populations of vessels. So you can, where do you get a brain vasculature map or geometry? You can get it from time, rotational angiography, from time of flight MRA, from contrast enhanced. Let's look for a minute just to two examples, 3D array and time of flight. They have very, very different resolutions. One is isotropic, the other one is not. They are used in different points across the pathway, the clinical pathway of the patient. So, you can do time of flight for follow-ups. You need to put the patient in a catheterization room if you want to do 3D array. So they're not exchangeable. Then you do images with those and you say, well, that's very easy to segment, right? 3D array, you don't see the bone, you just do a threshold and that's it. Well, it's not that easy. If you do thresholds, one user may get this vessel tree, the other user may get this vessel tree. If you have very close to the resolution imaging, you can have kissing vessels, missing vessels. And imagine if we're going to be looking into the fluid dynamics, all of those things are going to be changing the topology of our network and be, seriously problematic. So if you go, as we did in the past, and you look at algorithms based on things like level sets, I wish you luck, but it's very difficult to do it properly. And we've done lots of work and we got them to the point that we validated them, but it's very, very difficult to do that in ways that you can run it on any modality, on any type of data that's coming from different sources, for instance. So there's a lot of work of developing methods which are kind of all-terrain, right? Or as they talk in computer vision, to be operative on the wild, right? So very simple things we used to do in the past, like every step of this chain, like segmenting, computing center lines, computing the neck of the aneurysm. Now imagine doing that on a way that you can run it on tens of thousands of data sets without any manual intervention. So for instance, when we wrote that paper that I explained before, it required at least on the order of 6 weeks to do 82 cases, and there was a lot of manual operation. Now we have techniques that we can do it fully automatic within 30 minutes with all the steps. So what do we mean by all the steps? Well, not only segmenting the vessels, but segmenting the aneurysm within the vessel, segmenting the center lines, detecting automatically the neck, and all the entrance and output, the inflows and outflow points, so do that in a fully automatic manner, be able to automatically release the stent and get it to the, to be deployed without diverging, then do mesh generation with having good quality meshes, and then go and do your CFD. Okay, so all of those steps are things we do in our community. The challenge is scaling them up, and scaling them up, I think the solution is to incorporate priors there where we can. Other things that are problematic, so well, we do it with deep learning, perfect. But the problem with deep learning is, most of your clinical colleagues, if you're lucky, will give you a 3D segmentation of the region of interest of the aneurysm, and you want to segment the whole vascular tree. So we are working on methods in the lab where, we're given only a section of the image, and you can learn and extrapolate to the rest of the image with good accuracy. And then a lot of validation to be made in terms of, how parameters in the ground truths, in terms of, morphometrics of the vessels and the aneurysms correlate with those when you do that through the automatic techniques. Simple things like assigning boundary conditions. Boundary conditions depends on in which vessel of the circle of Willis you are. And that means that if I give you a perfect segmentation, you still need to do automatic labeling to tell what vessel is where, right? Which is the posterior communicating, what is anterior communicating. So doing that in an automatic manner is non-trivial, because the circle of Willis likes to have physiological variance. So these are all graph representations of different circles of Willis, all of which are physiologically plausible and healthy, but not all of us have the same topology. So it's basically a graph matching of a partial graph under multiple possible templates of graphs, so that you can then assign colors that represent certain segments to the vessels, and you can automate that. And then finally, we talk about big data sets and running it on thousands of cases, but the problem is that there aren't that many data sets that big, right? Or if they are as part of the patient records in hospitals, not necessarily in big trials or big population studies. Even studies like UK Biobank, for instance, that have at the moment on the order of 50,000 subjects that have been collected. That's as big as it gets. And imagine we take UK Biobank. First problem we find is that actually UK Biobank doesn't have a geographic data set. So you cannot use that to estimate directly the vessels. So what we develop is techniques that actually can synthesize vascular trees from the non-contrasted T1, T2, and proton density images, and generate synthetic trees. And just to make it a bit more interactive, I'd like you to think about this image. So this image has, one of these two images is fake, the other one is real. The one that is fake was generated from the real T1, T2, and proton density from the patient that we also had an angiography. So one of them is fake, but is the same, it tries to replicate the same anatomy in the vasculature that you would see. And I'm showing now here like a maximum intensity projection. And this is a Turing test, and I can tell you that it's challenging to figure out which one is the right one. So some of you, if you saw my talk in MIOA a few days ago, you will know that the synthetic one is this one. But again, it's difficult. It's difficult to figure that out. So that's the quality we can start achieving, and then all of a sudden we have potentially 40,000, brain vascular trees where we can run these trials. And a trial with 40,000 subjects doesn't exist in this domain. So how you evaluate then those synthetic data sets? Well, we looked into, for instance, not only evaluating accuracy in terms of morphological parameters that we extracted from the ground truths, but also in terms of, is our hemodynamics predictions based on the geometry from the synthetic data anywhere close to the hemodynamics that I would have estimated from the ground truths? And we, we've looked into that, and and it's given good results. I cannot go into a lot of detail about the method because there is some work being under review at the moment and some patent pending as well. But we do have the ability to develop those syntheses. So I like to, let me just jump for a moment. So I'm going to skip all the part where we were explaining that we did a lot of validation on the algorithms for fluid dynamics, and that we could utilize those simulations to create patterns. Like, we know that if the impingement size of the flow is very small, this tends to correlate with higher risk of rupture. While if the impingement zone at the end of the aneurysm is wide and gentle, it tends to correlate more with unruptured aneurysms. So you understand what are the patterns, the neighborhoods in the sat-nav analogy, the neighborhoods of the city where I don't want to get in because I'm looking for trouble. The neighborhoods in thermodynamics. And then you can develop recommendations that actually say, look, these are groups of patients that are higher risk than others, for instance. So this also links to stratified medicine. But we also did work, as I mentioned, in terms of interventional planning. And I want to talk a bit about the device modeling and what it takes to do it. So these stents that I talk about are very, very small. You can see that in the picture. And there is all kind of issues with them. The geometry has influence of the vessels, has influence on the outcome. The experience from the operator has influence on the outcome. Adverse configurations can lead to, problems downstream. So we developed techniques based on simplex methods and, work that colleagues in our community, in INRI and others have been developing hereditary jet for many, many years. So adaptations of those techniques and incorporating priors about the knowledge we have on the way these constructs behave allows you to do virtual implantations of those devices in a way that we model well the final position. Now, you may say, well, you can, how you evaluate this? Well, what we have done is evaluated the performance of these devices in a number of ways, but one of the ways is by looking at comparisons against the structural mechanics. So this is structural mechanics, for those of you who are perhaps less familiar with it. Our fast virtual stenting takes about a minute to deploy. The finite elements can easily take 8 hours. If you look at the publications, most of the publications in these areas are idealized geometries. When you put the complicated geometries that come from images, they tend to diverge. So we validated against some of those techniques working with people in Milan that are experts in structural mechanics. And the other way you can validate this is by comparing to generating a physical and a digital replica, where the digital replica is like a 3D printed, so the physical is a 3D printed version of the anatomy, and you put a real device, and the digital one is the virtual device deployed on the real image. And you can look at virtual angiograms and look at how, for instance, the bolus passing through these constructs. So if you forget about the blue, which is the reference point here, and you look at only pink and green, these are two volumes of interest in the same position. One is simulated, the other one is actually the time dilution curves from X-ray. You can see that they are pretty much spot on. And then you can compare, different scenarios, look at the sluggish flow, and understand how that performs in a number of anatomies and so on. So we've done the same thing with coils. Here is a bit more challenging because coils, you cannot really plan where you put them. It's like a stochastic. It's like an stochastic field. Just to give you an idea about the plethora of devices that exist, just on coils, I could show you the same on stents. There is tons of them. And each of those devices go through a 6 to 8 year cycle, testing on humans. I mean, imagine the cost, risk, everything that goes with it. So what we develop is a method that is based on minimum cost pass techniques that simulates the filling process and that we validated that by comparing that against histological specimens, where we can see the density of the struts and compare the density in the synthetic, in the simulated case, versus the density in a real specimen that they're matched. So for instance, you get things like this, where you can, each color corresponds to a different coil. Each column corresponds to a different percentage of what is called the volume filling. So 20% mean that 20% of the volume of the aneurysm is filled with metal, 25% filled with metal, 32% in metal. You see that it's very difficult to go anywhere much higher than 30-something%. And then each horizontal row, what it represents, if this is an stochastic process, it represents a different realization of that stochastic process. So configuration A at 20% is I put coils up to 20%, then I take them out, and then I put them again. They will go into a slightly different configuration. I generate configuration B. I take them out, I put them again, I generate configuration C. And you can imagine you can do that as many times as you want. The interesting thing is that what you can observe is that as the packing rate increases, it doesn't matter anymore how you actually put the coils. So all of these configurations, all of them converge to an asymptotic maximum flow that they're allowed to get through. And this is quite interesting, because what it does tell you is that if you put less than this critical point, you're likely to depend on what happens with the amount of coils you put. If you put too much more, actually you don't need that. You're spending more money, and it's about £1,000 each of them. And you're actually increasing the risk of rupturing the aneurysm by overpressurizing it from inside. So there is an optimal packing rate for every patient that you could plan. And interestingly, 10 years before we did that piece of work, there was a clinical study that showed that aneurysms that were stable after embolization, meaning they did respond well, tend to have a higher packing rate than those that were unstable. But also that the packing rate depended, the optimal packing rate, depending on whether the aneurysm was small or large, small neck with neck, large neck, which is a coarse version of the personalized packing rate prediction that we can now offer, okay? And then this can be validated again through comparing this with histology measurements and looking at densities of struts in the histology and density of struts with a virtual coiling in a digital twin version of that data set that we have the histology. Okay, I'm going to go back to the end now to the final conclusions, and is to say, look, there is tons of challenges for our community to contribute, whether it is on building virtual populations, building models of examinations and interventions, or developing virtual readers and virtual outcomes, which I haven't talked much today, but virtual readers is essentially AI, and virtual outcome is to finding, surrogate endpoints based on imaging, for instance. And that dream of, we don't need to be lost with the idea of a whole virtual patient. Sometimes it's just specific organs, but it can also be complex systems of organs, like the ESCAN Phantom that allows you to do sophisticated thorax models. This is not something that is a Star Trek. I mean, FDA is already publishing work where they have evaluated imaging techniques using in silico trials, and where they have shown that you can do that in ways that is cheaper, faster, better, safer, and more scalable. So everything you may want. And to conclude, I hope I convince you a bit, or provided more arguments to the fact that AI is making a lot of strides in healthcare, but that unfortunately we have focused a lot around purely automation tasks so far. And I think we need to start looking more towards going beyond that, and that the ability that models have to help to integrate and fuse multimodality data is great. And also that we need approaches that allows us to develop better algorithms that are all-terrain, that work on the wild, and that mechanistic-led AI together with phenomenological AI, both together actually provide a great opportunity not only to exploit the amount of data that we are increasingly acquiring, but also make sense of it in a way that can be that is generally valuable. And I'd like to finalize by saying thanks to a lot of other people. I only mentioned the academics in the slides, but there is a lot of very committed students and postdocs working on the group. And if you're interested in this area, we're more than happy to talk to you and perhaps have some of you working with us at some point in the future. And please don't forget about IPMIT that is coming up in a few months from now. Thanks a lot.
Thank you, Alex. Would you mind to take question on the from the chat room, or?
Yeah, absolutely. Or if people want to speak up, we can unmute them. Whatever works best for.
Okay. Maybe we can. Okay, Alex, thank you for your talks. Just before another audience asking question, can I ask one regarding to this this image-based simulation? You mentioned about this. Actually, the work seems to me involve these three very big topics. One is about like a fluid dynamics, and also the other one is because we have blood, we also have this solid tissue. So it sounds like there's some simulation works need to deal with about this interaction between fluid, blood, and the solid tissue like the vessel. And the finally one is like the image processing, like extracting the geometry of the vessel and the other tissues, which is basically I think is image segmentation. So from your experience to build up this massive system, how much effort need to allocate to this these three different parts, or how much credit you would share to this to reward to these three field in terms of building up this very complicated system?
So if I understand, Shahar, your question is, there seems to be some effort in what we could call pure image analysis, and other efforts as well in estimating mechanical properties. So more like, it's almost like image-based experimental biomechanics. And then there are aspects to do with numerical computations and predictions of quantities. And I think the question is, do I agree with that sort of division? And then what is the proportion? Yes, I think that is a good division. I think there is actually a lot more, because it's very difficult. In this particular example, for instance, there is also the biochemistry that was involved. I mean, there was a whole PhD thesis we did from Ali Sarami that actually developed models for clotting that can be utilized in this scenario. But I think, broadly speaking, you're absolutely right. We can use imaging either as an input to these models, whether it is from geometry, physics, chemistry, or as a validation to the models by comparing the outputs of our predictions with the output of the predictions with the things that we can image. And I think that's actually absolutely right. And therefore, there is a need to develop algorithms that can also help to do that validation. Let me give you an example. There's another piece of work we are doing on cardiac flow, where the measurements of flow that MR allows you to do in vivo, when you look at the voxel spacing, is actually very, very coarse. And when you compare that with the simulation that you can have in principle infinite resolution, it's a very simplistic validation, even if you compare with an image, right? So what we are looking is ways of doing super resolution on the flow based on the image, so we can have better image-derived information to validate against the model. And again, that's image analysis. I think there are a few other questions that are related to this in the chat that I'll try to answer in order. I think there is a question by Senius about what is the gap between simulation and the real world? What do you worry about in simulation-based trials? How do we control for this? Look, Senius, there is, in fact, I could say also, what do we worry with conventional trials? I mean, if you really think about what we do in conventional trials, a conventional trial is a model, and what I presented is another model. So they're both models. Animal experiments are models. Bench tests are models. So if we start by the point that these 4 ways of doing things are 4 models, there are models that are best for a particular scenarios and not very good for others, right? So for instance, when it comes to in silico trials, what I am concerned is about the ability to incorporate uncertainty as well from all our measurements. So I think we need to be reporting results of in silico trials, but also with the margin of uncertainty we have by accumulating, say, things on the uncertainty on segmenting, on simulating, on all the different steps. Now, there is also uncertainty in a conventional trial, but what an in silico trial allows us is to quantify it and put it up there. So the advantage of in silico trials, it forces to declare your assumptions and to make put up your assumptions, straight away. So that's what I'm concerned is how good our assumptions are, but at the same time, I know that's a strength because by declaring them, I'm forced to have to test them. What I worry about simulation-based trials, I think at the moment is having representative data sets so that we can build cohorts which are to all purposes equivalent to a prospective cohort that we need to recruit in a conventional trial. And at least in the UK, that is, I think, is starting to become easier because of the way that we have a national health system where things are federated, at least to some degree. I think national health systems will have a better go at this than if the system is very fragmented or very privatized or very regionalized. I think there is a question by Igor about the recording. I think the answer is yes, but you need to talk to Shahai for that. Zewan was asking if it's possible to compare the simulation results with in vivo results in animals. And the answer is yes, and I will give you an example. But also I will ask you to think why you really want that. Because ultimately, even if we can reflect perfectly an animal model, if the device will go into a human, I'm more interested in looking at, can I mimic the human conditions as close as possible? And I think we need to remember that an animal model, again, is a simplification is another model, right? But one example where they have done it, I know that Medtronic has, incidentally, these in silico trials are starting to be used in regulatory approval dossiers. I know at least, and that has been publicized, so I don't think I'm breaking any confidence here, Medtronic, a few years ago, passed through FDA approval a number of leads for Parkinson's disease. And as part of their dossier, what they did is, in addition to all the experiments that they normally do, they added another experiment that was looking at the heating effect of those leads in the brain. So the way they did is they validated the model on animals, and they compared the heating maps on animals measuring heating, I think they did it with MR, with a heating map of their prospective simulations. That's the way they validated their model in the way that Say is suggesting. And then what they did is they did predictions on humans by doing in anatomies of a human 1 million different possible configurations of the leads and ensuring that the heating map was below the safe level. And the person who was telling this to me was saying, look, and we in Medtronic have never yet implanted 1 million electrodes of those in the world. So it allows you to generate the scenarios and counterfactuals that would be very, very expensive to realize. Do you take fluid-solid interaction into consideration in your model? Maria, not in most of the things that I show, but this is because we did experiments in the past and we demonstrated that for the outcome measures that we are interested, that is not important. Fluid structure interaction is important if you're looking at endothelization, if you're looking at remodeling, but it's not so interesting if you want to look primarily to flow and to clotting, because this is largely a geometry-driven flow. How about the safety of virtual simulation? For example, the MRA synthesis. I'm not sure I understand what you mean by the safety. I mean, of course, synthesizing the image doesn't expose anybody to risk, but I suppose that what you mean is, what about if you synthesize a vascular tree that is not real, and then you make a mistake as a consequence of assuming that the geometry was correct? Remember that I didn't suggest this is a good idea necessarily for patient-specific planning or for patient-specific diagnosis. What I suggested this as a way to generate virtual populations. In a virtual population, if you have a very, very large set and some of those geometries are a bit kind of less plausible, let's say if you think this as a distribution and you go out of your main bandwidth of your Gaussian, what you're effectively doing is over-testing your device. So as long as you make sure that you represent at least the plausible cases, at least the fact that you have a bit more, that you have some additional cases that are less realistic, I think is a bit less of a problem. Yeah, there's a question by Heiko. I would be very keen to follow up perhaps with you, Heiko, on that. But yeah, you're absolutely right. And the other thing that is very important is, imagine we are starting to talk a lot more about uncertainty in image analysis, right? But imagine in a world where all of us publish algorithms without providing the uncertainty, how are we going to be able to estimate the uncertainty of the pipeline, which is a composition of all of that? So that's also an important element. Fuping have yes, so I think Fuping's point is about, and Heiko perhaps as well, is about when you do an in silico trial, that is a sequence of operations. One thing that I think we need to do more in our community is over the last 5 years, perhaps, there has been an increased interest in the problem of quality assessment of the images, but also quality assessment of the results of the algorithms. I think in the future, every algorithm that we develop, we need to declare what are the inputs, what are the outputs, the mean errors, the uncertainty on the output as well, but also we need to have some sort of flag that says, I'm failing, I'm not delivering a reasonable result. Because if you would have that, what you could develop in these pipelines is a mechanism to understand the whole system, where it fails the most, and how you need to bootstrap the design. And the other thing is, do we care that we have 100% accuracy, so not 100% accuracy, but 100% success rate, that every image we put at the input perfectly comes out at the output? I would argue that we don't, that we have very large databases where we say only have 90% success rate, but we can tell which is the 10% that fails, so that you can assess the biases by discarding those. But discarding those, if you have a very large number at the beginning, it doesn't matter that you lose a few patients. This is equivalent to the attrition rate in patients in a conventional trial. What is important is that you're able to qualify and quantify that attrition rate so that you understand the impact it has. And if the synthesis tool can be used for other modalities, yes. And I would even say more. We have a paper published earlier this year where we show that you can do even cross-organ synthesis. So we have a paper where we look at eye scans and you can actually do synthesis of cardiac MR images from the same data set. That's what we did with Marco Lorenzi and some other colleagues. And I'm very happy to point you to that. That is published and I think we need to develop better algorithms. I'm not pretending we have solved ourselves the problem, but what I'm saying is I've seen results which are actually quite encouraging that this is feasible. And there is a very interesting paper by Jong Chul Hee from Korea, I think in Nature Machine Intelligence as well, where his group demonstrated that you can synthesize any endogenous contrast agent from MR. So if you have an imaging, an MR image protocol, you can synthesize other imaging protocols from MR as long as those other images are endogenous contrast agents. So, you know, time of flight, phase contrast, but not things like gadolinium enhancement, which seems to indicate that all of these are like different projections of fundamentally the same information. And that is what, from a theoretical point of view, encourage us to do the synthesis work that I showed you before. I hope I covered most of the questions that were raised. Otherwise, very happy for you to drop me an email, and I'll put my email on the chat. And, you know, very happy to explore ideas in these areas with any of you. Very good. So I think I'm probably standing between you and your lunch or whatever it is in your time zone. But thanks a lot also for the questions that make it even more enjoyable as well too.
Thank you, Alex. Thank you again, and thank you for all the audience for coming. And we will also announce the next coming fellow talk soon, and hope to meet you again online next time. Thank you, Alex, again. Thank you.
Super. Thanks, and have a good day. Cheers.
You too. Bye.